forget. So because I'm recording, it's not going to be a good lecture. The two lectures I've given this semester that were really good, I both neglected to record, or at least that I thought were really good. Okay. So we will have an extra assignment in here, an extra, I don't know, we're going to grade it under quizzes, under exams, I guess, but it's not really going to be like a traditional exam. So let me show you where it's going to come from. So if you are feeling industrious, you can read ahead. So if I share my music. Okay, there we go. I've become a big fan of this NORD website, the National Organization for Rare Disorders. They pretty much, if it's an unusual disease, they try and catalog it here. So the one, just by happenstance, someone I know happens to have a child who's been diagnosed with this KCNB1 encephalopathy. And how rare is this? Well, it affects, it's known to affect about 65 people in the world. It's only really been identified in 2014. So, there's not a lot going on with it. But the interesting thing is this KCNB1 gene encodes a potassium ion channel. So since we've been talking about ion channels, I was like, all right, let's get some practical knowledge about this. So there are a couple known mutations that end up causing a defect in the channel. And I'll let you guys read about this, but I figure there's not a ton of information here. But I have devised probably 5 or 6 questions. So I'll have you guys read this and then have a short timed exam for you guys to come up with answers to that, which will help bring up any exam grades that you may be concerned about. So there we go. It's not really extra credit because it's a graded thing on its own, but it gets us used to start applying some of the information that we've talked about. And that's the tricky thing about teaching biochemistry. There is a ton of foundational information in here. And then you have to start applying it. But in order to apply, you have to cover so much information. It's the same way in biochem too. When you talk about metabolism, there's so much going on, and then actually use it. So I've kind of tried shifting a little bit of my I'm going to say the bad word, pedagogy, towards the application at an earlier level, so that you can get used to seeing why it's important to know this. But so like I say, I'll have a link to this particular disease on this website, and maybe 1 or 2 other links if you're interested, but that would be something to kind of look up in there as well too. Again, this is not a scientific journal article. This is just kind of a straightforward report on it. I talked about different therapies and other stuff that are in here. And it actually does give some nice references in here as well too. So either one of these papers would be potentially worth looking at. Anyway, that's what's coming up. So let me share this. So look for that later this week. And then, like I say, we've got 1 more exam, probably geared for next Tuesday. And we have 2 more class periods to cover for 1 more chapter. So yay. We're on schedule, which is actually the first time in years, because usually I'm way ahead of schedule because, I talk so fast, right? Cover so much information. Okay. So let's take a look at our chapter 12 slides. So here we are moving from our cell membranes and transport into biosignaling. And we might as well call today the GPCR lecture. But we're going to start with a little background on biosignaling, which if you have a really good memory, you may remember, hey, we saw one of these slides in like chapter 2, I believe, 2 or 3. I think it was 2. All right. So we're going to talk about signal transduction. So now we've got all of our proteins, our lipids, our carbohydrates, and our nucleic acids, and they're all contained within a cell, and now they have to communicate. So we can start building up to a multicellular organism, even within a monocellular organism. Um, they still have to have signal transduction there. Take information from the environment and get it to the nucleoid so that the cell can figure out what it needs to do. Right. So we'll talk a little bit today about signal transduction and G protein coupled receptors, and on Thursday we'll finish up with the other types of common signal transduction mechanisms that are in there. All right. So if we get signals, for example, I woke up today and it's cold, so I'm like, oh, better wear a sweater. I've got my environmental signals in here. All right. If you think from a cellular perspective, we can have antigens that will bind to the cell surface receptor. We can have hormones that can either bind to the cell surface receptor or be taken in and bind to the nucleus, to a nuclear receptor. Neurotransmitters, even things like light or touch. If you think you wake up in the morning and open your curtains and you see, oh, sunlight, and your pupils will contract. Okay. Um, you know, um, Pheromones function this way as well too. But all of these signals are going to cause changes in the cell's function and potentially composition. So we can see things like antibody production, right? Or maybe we can something will signal our cell to grow, or atrophy as well too. We can talk about cell division as well. So all of these are dependent upon cell signals that come in. All right. So we're going to talk about some of the cell signaling mechanisms, and we're just going to simply take our receptor as some type of membrane-bound or soluble protein or protein complex, which exerts a physiological effect. So we're going to call this our intrinsic effect, a physiological effect after it binds its ligand. All right. So we have 5 main ones that we can talk about here. 4 of them are in the membrane receptors, in the membrane, and 1 of them is in the nucleus. So we're going to talk mostly about G protein coupled receptors today, and we're going to talk a lot. We're going to get into a lot of detail about GPCRs. Then we'll talk about things like enzyme linked receptors, ligand gated ion channels. So again, we will be focusing on that KCBN1 channel for an assignment coming up. And then we'll talk about integrins, which are probably one of my favorites. And then finally, we'll talk about nucleoreceptors. All right. So our receptors, whether they're in the nucleus or in the membrane, are going to bind some type of ligand. Okay, these can be small ions, organic molecules like adrenaline, polysaccharides, peptides, or even other proteins. Okay. So again, we're binding something, and then we're triggering an effect. Okay. So in order to have that signal transduction, there are 5 features. And this is how I know I'm getting old, because it's like, well, actually we got 6 listed here, don't we? A, B, C, D, E, F. It's hilarious. It's hilarious because when I started teaching, earlier editions of this talked about the 4 features of signal transducing systems, and they had 5 things listed. So I don't know. Crazy. Anyway, what I like is to make up a mnemonic to help keep these straight. Okay, so we're going to talk about these 6 things, and then we're going to come up with a mnemonic for it. All right. So the big thing is, our receptor has to have specificity. It should only be able to bind a specific ligand. Now, some receptors are a little more promiscuous than others and can bind classes of ligands or multiple ligands. But for signal transduction, we're normally going to see, 1 or 2, maybe a very small number of ligands that can bind. So that's our specificity. And here we can see we have, signal 1 will bind here. Signaling molecule 2, nope, can't bind, won't fit. Okay. We're also going to see amplification. And this is the big thing here. When our initial molecule binds, we're going to see an effect that causes this amplification. So we might have 1 molecule that binds here, which triggers a release of 3 molecules, which each trigger 3 more. And after a couple rounds of this, we have this 1,000-fold or 10,000-fold increase in reactivity. I think we sort of saw this, or maybe we'll see this again when we talk about glucose release. We can have 1 molecule of adrenaline that will bind, and it will trigger the release of 100,000 glucose molecules in a very short period of time. And that's that whole massive adrenaline response. A bear jumps out and attacks you, or something like that. Or a clown. I don't know, people are afraid of clowns for some reason. But amplification is going to be our second feature in here. We also have this modularity, okay? And we're basically talking here about signaling complexes that can form, all right? So we can see multiple proteins and molecules that can bind together into a complex. And then sometimes all we need to do is change out one complex and we can get a different signaling pathway. This modularity actually helps tie in with integration, okay, which we'll talk about here in a minute. So we can basically form these complexes with different pieces for our specific signaling response. Okay. We also have something called desensitization or adaptation. If we were in 319, did I say 319? Yes, 319 in WBA. I would use the example of the air conditioning unit up there because whenever I walk in that room, I can hear the machinery in the air moving through there and that little kind of electrical hum. But after I've been there for 10 minutes, I don't notice it anymore. So that becomes kind of an adaptation response. One of the things I studied as a postdoc were the ear hair cell cells in the ear that are responsible for hearing. And essentially there's, all these different little hair cells that kind of wind around in a spiral pattern. Each one is a little bit different, responds to a slightly different um, um, noise wavelength in there. So that is responsible for our hearing, so we can hear those different signals. But then, after the noise comes, we trigger this response in there, and then it becomes desensitized, it adapts and recharges so we can do it again. So again, if you have this ongoing hum, you become desensitized to it. Ideally, so that you can have your response and then kind of say, okay, I heard you. It's kind of like, I don't know if anybody's ever been in a relationship. Yes, I heard you the first time, you know. So our desensitization there too. All right. Okay. So going back to connecting our modularity with our integration, we might have two different signals that come in that are going to trigger two different effects, but somewhere downstream from these initial two receptors, there's going to be what we call crosstalk. Okay, these two pathways can talk to each other. The example I always like to use in biochem 2 is a bear jumps out, you get this massive adrenaline rush, right? So you're mobilizing, all your glucose, all your energy there. Okay, but suppose you just drink, a giant Pepsi and eat a whole bunch of leftover Halloween candy and consumed a bunch of sugar. So your body's in a well-fed state. So you've got one signal there saying, oh, we are energetically favorable, we're well-fed, let's store this glucose as glycogen. And then, a bear jumps out, and you get this adrenaline rush. You're like, oh no, release the adrenaline. So you have two conflicting signals. How they resolve is going to depend on the integration pathway in here. And generally adrenaline takes precedence over storage. So yeah, you have a lot there. And then the newest thing here is we're going to have a localized response. Okay. So, we've got an enzyme in here and we have this internal message. We're going to have something that's going to say, okay, let's keep this local. It's kind of like all politics are local. It's like everybody gets all up in arms about who they're going to support for president. And, I've known in my lifetime, maybe like 5 or 6 people who have been directly affected by a policy of a president, but on the local level, I've known hundreds and hundreds of people who've been affected by some policy that's changed in the county or in the city or something like that. So our localized response is another feature in here. We don't want to have a signal in the kidney that also gets shunted over to the liver or something. Usually, it depends on our type of signal as well too. So these are our primary features. So how to memorize these? All right. I look at this and, if I just take this straight forward, I'd call it Samdil. Yeah, it's like he's in Lord of the Rings or something now. But we could also, just use this to spell something. I used to go with MAIDS, MAIDS, the five, the MAIDS of the transduction signaling system in here. If you put in the L for localized response, or maybe an R. Maybe you call it marids for a type of Arabic genie. I believe they were. What were the marids? Were they the water-based genie family? I can't remember off the top of my head, or maybe air. But anyway, you know, some type of mnemonic like that can often help. All right, so it gives you, specificity, amplification, modularity, desensitization, integration, localization. I actually prefer adaptation to desensitization, but it's easier to spell with a D in there. But anyway, that's just some suggestions, ways to kind of keep things um, in your brain. Right? Um, we have there. We can actually have an. I don't know. Anyway, sorry. I'm trying to spell things with my abbreviations. All right. So again, specificity, just to kind of go over this, we're going to typically look at maybe a typical KD is going to be something like 10 to the negative seventh or lower. So we have, basically one binding molecule to these signal transduction receptors. Okay. So let's talk about some common membrane receptors, and we're going to start and focus primarily today on our G protein coupled receptors, or GPCRs. All right, and I will always mix up, for memorizing, like the 5/6 features of the signal transduction pathway. I always mess up GPCR, and I'll call it GCPR at some point. I just know I will. Anyway, our G protein coupled receptor involves something called a G protein. Ooh, imagine that. Okay. And this G protein is the trimeric protein that can bind a ligand. And when the ligand binds, we actually see a separation of one of the components of this G protein complex, which will then move to a nearby molecule to activate its signaling pathway and start amplifying it, usually to make a second messenger. We'll get to that in a second, okay? So we're gonna generate the second messenger. We can see this moves over here, okay? We do have others besides the G protein coupled receptor. We have these tyrosine kinases or receptor enzyme kinases. So basically we have, something here that looks like, maybe chicken legs or something. It's the bottom of a chicken, all right? Our ligand binds to the receptor, and we induce phosphorylation. So we have this kinase cascade. So we'll see, maybe this will activate MAP kinase, which activates MAP kinase, which activates MAP kinase, and so on and so forth, until eventually you have all these things that are phosphorylated, and that can result in a nuclear signal, okay? We can have gated ion channels. So these will open or close when a ligand binds, which will allow ions to flow in or out, okay? And when that happens, we depolarize the membrane, which results in a signal that's occurring in there. Not shown on here are my favorite ones, the integrins. They are basically connected to the cytoskeleton and the extracellular matrix, so you can actually get physical tension that will cause the integrins to induce a conformational change, which when that pulls on the cytoskeleton results in a signal. I kind of think of them as like telephone wires in a way, and they're transmitting across the membrane there. And then finally, we can have nuclear receptors. These do not bind to any membrane-bound receptors. They go directly to the nucleus. They do not pass go, do not collect $200. I don't know if anyone plays Monopoly anymore. But they go directly to the nucleus and bind to binding partners in the nucleus, which can then bind to the DNA and induce gene transcription, or turn it off as well, to turn on or turn off genes. Okay. So our G protein coupled signaling is very well studied. The general mechanism is quite well known. Our G proteins are heterotrimeric, so that means they have three different substituents, an A, a B, and an alpha, a beta, and a gamma protein that come together as this small complex, and they bind GTP. Okay, so that's probably where the G protein comes from. All right. Our G proteins will mediate a ton of signal transductions, and they mediate them to other target proteins in here. So rather than directly targeting this enzyme, like adenylyl cyclase, we target this G protein, and then the G protein activates that. Why that two-step? Isn't that kind of inefficient? Well, this actually helps with amplification. So that whole amplification that's in there. We can also have, a large number of adenyl cyclase molecules in there, and we only need a few G protein coupled receptors to trigger a big effect that way. And then we can have different variations of our G protein coupled receptors. I think there are several hundred to several thousand GPCRs that are out there that each have their own specific binding ligand. All right, so that way we can have a wide range of reactions. Okay, so our typical G protein is going to be something like the RAS molecule. We have an integrated magnesium in there as well. And then here's our GTP that's bound in there, and the magnesium again, just like we see magnesium with ATP, helps stabilize those phosphate groups that are in there. Okay, we don't need to worry too much about this, but in effect, our G protein is kind of acting like a switch. We're going to see what's going to happen here. Okay, so I talked about adrenaline. Epinephrine is very similar. Epinephrine will interact with the cell through a GPCR mediated response. Okay, so basically it's our stress response in there, and you get a shot of epinephrine. It's like, oh, yeah, you get more energy. Okay, but it also helps to alleviate stress, or, well, I should walk that back. I was gonna say it mediates the stress response. Maybe you can, and some people can enhance it. Like an allergy medication that says it can cause drowsiness, and then in some people it causes hyperactivity. I was a kid that it caused hyperactivity in, so kind of an antagonistic response. Might need to get a drink out of my little fridge over here. Another good reason to teach from my office. Okay, but typically our epinephrine will bind to receptors in the muscle or liver cells and induce the breakdown of glycogen. Basically, epinephrine is released in response to, hey, the body needs energy. Release the glucose. Right. It'll bind to receptors in the adipose tissues and induce lipid hydrolysis. Release the free fatty acids. All right, and it'll bind to receptors in the heart and increase the heart rate. So we can play the dramatic music. Maybe I should have started more dramatic music for my opening thing there too. Okay. There's a neat comparison in here. If anybody's thinking about pharmacy or pharmacology, epinephrine, this is its structure. There is an agonist, isoproterenol, which binds, about 10 times better. And I believe this is an anti-anxiety drug. No, maybe this is one. No, this is an agonist. Okay. So isoproterenol will actually function like epinephrine. So, but it's 10 times better at binding. So, it basically has the same effects as epinephrine. And you can see its structure in here is very similar. Propranolol, which, let's see, I forget what all that's prescribed for, but I know one is an anti-anxiety medication, and it binds, 100 times, 1,000 times better than epinephrine. And counters this stress-mediated response. So I know some people who have been prescribed propranolol for anxiety or something along those lines. It also has another effect, which is escaping me off the top of my head here, and I don't know if anybody who's actually working as a farm tech or something. I have almost every year I've had somebody as a farm tech who, I don't need to remember what propranolol does because somebody in the class actually is like, oh yeah, because it's a fairly commonly prescribed medicine, but it's got at least a couple things that it's used for. But again, you can see its structure. Clearly, this ring structure here is going to play some type of role, and then maybe this ending up here, or actually maybe this hydroxyl group is one of the more important things to allow to fit in there. But again, these have structural similarities. The neat thing is, here's our natural molecule, natural ligand. Here is an agonist, and here's an antagonist. Okay, and the differences between them are really not that big. But we can see we can have greatly enhanced response and a greatly antithetical response just from small changes in our structures. All right. Anyway. So those are our ligands. All three of those, so our isoproterenol and our propranolol would be competitive inhibitors. They would bind at the same spot as epinephrine, but our epinephrine binds into this little specific spot on the outside of the G protein. I believe this is the beta subunit that has the transmembrane region in here. When that binds, okay, oh, nope, sorry, that's our receptor, which is coupled to the G protein. So yeah, so this is just our receptor. Here we can see our alpha, beta, and gamma subunits. Once this ligand binds, it's going to induce a change that's going to cause the G protein that you can see is loosely associated on the inside of the membrane to kick out the GDP and bind GTP. Okay, so once it binds GTP, then the alpha subunit is going to leave and go find adenylyl cyclase, right? So it's going to leave behind the beta and the gamma subunits that are still in here. You can see we still have this anchor for our alpha subunit. So it's basically going to slide along the membrane until it finds adenylyl cyclase, and then it's going to activate adenylyl cyclase. Adenylyl cyclase will then bind ATP and convert it into cyclic AMP. Okay, cyclic AMP is an example of a second messenger. Okay, so our, in this case, epinephrine is our first messenger. Our cyclic AMP is going to be our second messenger. Cyclic AMP actually can do a whole bunch of stuff, but in this case, it's going to activate protein kinase A, which will phosphorylate a bunch of cellular proteins, which will cause a cellular response, which will be that release of epinephrine or that increase in heart rate or that hydrolysis of lipids in there. Okay. Eventually, cyclic AMP will be degraded, which will reverse the activation of protein kinase A. I believe it's phosphorylase B, which will then start dephosphorylating proteins and get that adaptation response. We also see that the alpha subunit of the G protein will slowly hydrolyze GTP into GDP, so it will release from the adenylyl stop activating it and roll it back over here into the complex. Uh-huh. Alrighty. Okay. So, where were we there? Yes. Okay. So that's a lot on this slide. This is a whole lot going on here, and we're going to talk about some of these individual responses. But we basically have this big step, and there's even more to this. But I don't know if it's a good thing or a bad thing. We know a lot about our G protein coupled receptors, and there's a lot of ways we've seen them activate. But that's a lot of information, so that's why we tend to focus on this a whole bunch. Okay, but let's go back up here and look at our second messenger, our adenylyl cyclase. Okay, once our alpha subunit with its bound GTP arrives to it, it's going to convert ATP into cyclic AMP. So we get the energy from hydrolyzing our pyrophosphate here, and we're going to connect our remaining phosphate group 5' to 3' within that ribose ring of adenosine. Okay, so now we have our cyclic AMP. This can be hydrated, all right, to produce just AMP, regular AMP. Well, again, just our phosphate here is. So our cyclic AMP will function as a messenger in water. Eventually, it will get converted into AMP, and that will cause it to fall off of its protein kinase A and adapt or desensitize the response. Right? So cyclic AMP, very common second messenger. It's actually going to do a whole lot of stuff in the cells. Okay, so we see this amplification. All right, so all we need to do is activate just a couple of these G protein coupled receptors, and that will activate several adenylyl cyclase enzymes, which will release cyclic AMP and activate a whole lot of protein kinase A, which will activate thousands of glycogen degrading enzymes. So you get tens of thousands of glucose molecules released in the bloodstream. So I like to tell my personal example here. Is shortly after I got my new car, which is not my new car anymore. It's like 4 years old now, something like that. It's several years, three. I've had it for 3.5 years, I think. But it was new to me. Shortly after I got it, I was driving near Tanglewood, and there's a little road where it turns in. There are 2 lanes in there, and basically, I think a car that was turning didn't see my car because a car in the other lane was blocking it. So he turned right into my path, and there was no stopping. So I'm like, I got the kids with me, so I'm like, so I get that massive adrenaline rush, and I like my new car because I managed to steer between him as he was slowing down and a car that was starting to pull out the other way to turn, but who saw me had stopped and missed both of them by about 3 to 6 inches on either side. Um, but it was a really close call. And anytime something like that happens, bam, you get that adrenaline rush. In this case, similar to our epinephrine cascade, and just this massive release of energy. And so you're really kind of up. I don't know if you can see me here, I guess. And then what happens after that? A few minutes later, you get the crash, right? Because the adaptations coming into play, all that cyclic AMP is being hydrated. There's no continual release of adrenaline, so you're not activating more of those GPCRs. You're not making more CAMP. You get that rush, that quick rush, and then the crash as you now have all this glucose in there that the fight or flight mechanism. Okay, I don't need anything, so you kind of get that little crash feeling that coming down as the flooded glucose. It's like, okay, now we got to clear the glucose out of the bloodstream because we've made more than we need. Okay, all right. So that's kind of a physiological example of our signal cascade. And of course, something like this, you can see why that rapid amplification is important. If you're in like a close call or a bear is attacking or something, you want to have that energy made available immediately. If it takes you like 30 seconds to get all that glucose, well, you're already dead or maimed or whatever. So fast is good. All right, and probably should have had that picture on here as well too. 1 molecule of epinephrine can activate 10 maybe G protein. So we get 10 molecules. Anyway, we can see this. 100,000 glucose gets released very, very, very rapidly. Okay, so why do we spend so much time talking about G proteins? Because our second messenger, cyclic AMP, activates protein kinase A, and protein kinase A will activate a lot of stuff. It can activate on tons of stuff. And here you see our serines in glycogen synthase, or phosphorylase B, or pyruvate kinase, or hormone-sensitive lipase. Again, we're seeing a ton of serines and an occasional threonine down here in protein phosphatase inhibitor that will all become phosphorylated. So again, those hydroxyl groups on serines and threonines are excellent targets for phosphorylation. And we're essentially turning on or turning off a whole bunch of genes. We're turning off the ones that are going to be storing glucose, and we're turning on the ones that are going to be mobilizing it. You can also see this cardiac pump regulator as well. We're going to phosphorylate that so we can modulate our calcium levels, so you can get that heart rate up, so you can get that oxygen, again, all this stuff that's happening here at once. So again, our one particular epinephrine GPCR, our epinephrine receptor, we're going to see all these things are going to be modulated based on just that one particular receptor. Okay. All right. So let's talk about that crash, the sensitization. Okay. So epinephrine is meant to be a short-acting signal. If your epinephrine is constantly active, and I think that's why there may be a genetic, I don't want to say, try not to say defect, but a genetic issue that causes high anxiety. So that may be why some people are prescribed the propranolol. So it's supposed to be a short-acting signal. You don't want to always be releasing the sugar and kind of, always on and twitchy. Maybe unless I'm trying not to make meth jokes. Okay, and so we have to stop the glucose synthesis if there's no more need to fight or flee. Okay, so again, if we have high levels of glucose in the bloodstream, well, that can lead to diabetes and inflammation and all sorts of stuff in there too. So we want to have it mobilized for that fight or flight response, but then we want to clear it so that we can maintain the overall health of the organism. So again, we're going to see this desensitization or adaptation. Okay, it's like, oh my goodness, it's a bear. And it's like, oh no, it's not really a bear. It's just, my neighbor's got rugs out or something. I don't know. It's a clown. Okay, no, it's just a leftover Halloween decoration. Oh, it's a politician. There we go. All right. So there's a couple ways we achieve this adaptation. All right. The first is self-inactivation. All right. So we get our response. Our alpha subunit binds GTP, and then that dissociates from the beta gamma subunits and travels over to adenyl cyclase. But the alpha subunit, given some time, will hydrolyze the GTP back into GDP and release inorganic phosphate, which will cause it to stop interacting with adenyl cyclase and slide back over and reactivate with the beta gamma subunit. Mnemonic for this, it's like, I just can't quit you. I don't know, make a Brokeback Mountain reference here or something. It's weak. Whatever works to keep things in your brain. I would share some of the ways that some of the mnemonics I use in biochemistry to remember, especially metabolism. But I'm sure I would get complaints and violation investigations and stuff because they were just filthy, just awful, horrible things. But I'm like, oh, okay, I can memorize this by associating it with terrible things because apparently Dr. was a terrible person on the inside. Anyway, so this auto hydrolysis of GTP is part of the desensitization process, okay? And it becomes inactivated. We got our little flash lines there. I forget what they call those in cartoons. There's a particular term for those little lines in cartoons that are showing, activation. What that's, it's stolen from the comics pages and put into biochem. But once we hydrolyze our GTP into GDP, it becomes deactivated, slides back over, rejoins its beta and gamma subunit, and is ready to go for the next signal. It's like, oh, another bear. It's a bear dressed as a clown. See today's theme. Okay. There's also another way to do this as well too. We can have epinephrine binds to a beta adrenergic receptor. Okay, so what we're going to see is that there will be a bark protein. So once our alpha subunit leaves, our bark beta adrenergic receptor kinase will now be able to bind to the receptor itself. And when it does that, because it's kinase, it's going to phosphorylate it. Once the receptor is phosphorylated, it will attract this beta arrestin, okay, or bar, and this beta arrestin will trigger the endocytosis of the receptor, okay, and will bring it into the cell in a vesicle. All right. So basically, if you've got epinephrine that's being released, we're like, I had a joke for this, but I can't remember. But it's like you can't activate the function if there's no receptor, so you just bring the receptor inside. All right. It's kind of like what is it? I guess like coaches tell a lot of football players, like, no reason to be out after midnight. You can't get in trouble if you're at home in bed, usually. We're just going to bring the receptor inside. If the receptor's inside, epinephrine can't bind it because epinephrine can't cross the membrane by itself. All right. So that's the second method of adaptation. Sometimes, maybe we can degrade the receptors, but a lot of times they're just kind of held in here for a little bit of time, and then the vesicle goes back to the membrane and releases the adrenergic receptor back into the membrane. Okay, so that's a second adaptation response. Okay. All right. So, again, going back to the cyclic AMP, it's a very common secondary messenger, and there's a huge number of G protein coupled receptors that mediate their effects through cyclic AMP. All right. A lot of these will activate cyclic AMP synthesis, but some of them have the opposite effect. They will inhibit cyclic AMP synthesis. Okay. The neat thing is there are at least, somewhere around 1,000 of these 1,000 different G protein coupled receptors out there. Their ligands can be hormones, growth factors, neurotransmitters, all sorts of stuff. So basically you have all sorts of different receptors and different associated G protein coupled receptors to go with those receptors. Okay. Um, if you think about taste or smell, your olfactory and whatever taste is, I forget the Latin derived term there for that. But those are based on G protein coupled receptors as well too. The neat thing is, you can build a career here. There's a bunch of GPCRs that we have gene products for. We don't know what they're coupled with though, so you can go fish and see what they are coupled to and find their ligands for their receptors. Okay. Here we see a lot of things that are involved in using cyclic AMP. Tastants, I guess that's in there. Somatostatin, serotonin, many odorants. Glucagon is probably the big one because glucagon is kind of our antithesis of epinephrine. Epinephrine is like, hey, we need energy. Glucagon's like, yeah, we got plenty of energy, store it. So, all right. It's like two competing foremen trying to load and unload a boat is what I think about there. All right. So cyclic AMP can mediate lots of signals due to the particular localization of protein kinase A. All right. So the way we draw these pictures and the way we think of ourselves, oh, they're big bags of water and everything's just kind of floating around in there. Really not true. That cytoskeleton is omnipresent and kind of directs everything. Just like we have membrane rafts in our membrane, little localized areas, specialized areas within the membrane, we can have specialized areas within the body of the cell as well too. So our protein kinase A can be localized in particular regions by an anchoring protein, and that anchoring protein can interact with various components in here. So whether that is something like no, this does not seem to be the right picture, but here's a beta adrenergic receptor, and here's our rhodopsin receptor. Very similar. Rhodopsin is actually, I believe, light is its ligand in here. You can see arrestin bound to it as well. Okay. Anyway, there are some bacteria that produce toxins that will inactivate G proteins. So cholera and pertussis both function this way. They will inactivate a particular G protein coupled receptor. I know with pertussis, you get the that very wet cough because you're producing so much mucus because you end up blocking this G protein coupled receptor, so you get this buildup in there. With cholera toxin, what happens here is that you have this ADP ribosylated G protein. So it makes the GTPase constantly activated. So you will always have adenylyl cyclase activated, this constitutively active. We tend to think about defects arising from when we break something, we turn it off. But if, and what cholera is doing is essentially preventing us from turning it off. So it's always on, okay? And that results in the disease state there from cholera. Okay. We talk about cyclic AMP a lot because cyclic AMP is used so much, but there are lots of other secondary messengers out there as well too. For example, cyclic GMP. Okay, that's really creative, right? Instead of having a cyclic AMP, we use cyclic GMP. Yeah, about as close there. But inositol triphosphate, IP3, and because I'm like 10, it's like IP3. You remember that author of the book there too? But even calcium. So if we can have our G protein activates, say, a calcium channel that increases or decreases the influx or efflux of calcium in here, or activates calmodulin, we can see this calcium that will be released, and in this case bind to protein kinase C. So our calcium itself is functioning as a second messenger. Okay. Our IP3 comes from phosphoinositol diphosphate, okay, which is a membrane-bound phospholipid in here. It cleaves that to inositol triphosphate and a diacylglycerol in there. And that can bind. And you can see here, here we are making our G protein activate this. Our IP3 binds to a calcium channel, activates calcium channel, release the calcium. This activates protein kinase C. And, as opposed to protein kinase A and protein kinase B, which have their targets. Protein kinase C is going to have its particular targets as well too, and activate them through phosphorylation. Whew, that's a lot, right? It's a lot about our G protein coupled receptors in here. Okay, and again, our calcium channels and so on and so forth here. So besides protein kinase C, there's also calmodulin, which clearly is, nice names for these, that clearly involved in modulating calcium levels. All right. Anyway, okay, so here we have an example of just some of the structures of calmodulin. We're not going to go into structures. I don't want to murder your guys' brains any more than I already am, so we won't sweat about that too much here. But the thing is, calcium regulation plays a very large role in the body as well too, just like we saw with our cyclic AMP. Here we have, cyclic AMP is our second messenger, but we can also have our calmodulin regulating calcium dependent channels in here. That might be a good question, is, does calmodulin play any role in that KCNB1 that you'll see here? And I'm not sure because I haven't looked that far into it yet, but I don't know. I'll ask you questions I don't know the answers to, at least yet. All right, but you can see there's a lot of calcium channels and calcium pumps and such in here. I always like nitric oxide synthase as well too. All right, continuing on, our GPCRs, I know we mentioned they have functions in taste and in smell, but also in vision. Okay, so here's our rhodopsin receptor. We can see our rhodopsin. We convert our cis-2 trans retinol in there. All right, 11 cis retinol gets converted to all trans retinol through a photon hitting rhodopsin. That triggers the response of our G protein coupled receptor. We bind our GTP, release our alpha subunit, and it goes and activates phosphodiesterase. Okay, and when it binds, it's basically kicking off the inhibitory molecule. Okay, pulls off the inhibitory molecule, and then our phosphodiesterase gets activated, and it converts GMP into cyclic GMP, right, which can bind to this cation channel, causing it to close. So we don't allow our sodium and calcium back into the cell. So we're going to polarize this membrane, and this polarization is going to get transferred to the brain. We have this continued efflux of calcium, so we're building it up on the outside and taking away from the inside. So we're reducing that calcium levels in there. Eventually, when we reduce the calcium levels in here, as we decrease that, we're going to see GTP get converted to cyclic GMP that can activate another pore in here and allow calcium to flow back in. We also have a rhodopsin kinase that will bind to our rhodopsin with its transretinol in there and phosphorylate it. And then, of course, we can bring in an arrestin, which can bring that into the cell. And then the arrestin will dissociate, and the transretinol will be released and replaced with our cisretinol, and then we can do this again. All right, so we can continue this over and over and over again. We are now ready for another photon to come in there and activate it and get through the whole process. Okay, so what do we have here? Yeah, all right. So the question that you're probably thinking now is, what in the hell do I take away from all this? You can ask on the exam. So everything is fair game, and it's pretty much the bulk of this lecture is on G protein coupled receptors. So it's basically how they work, and the big key is our ligand interacts with the receptor, which triggers the replacement of GDP with GTP in the alpha subunit, causing the alpha subunit to dissociate and go find its particular binding partner and activate its binding partner. Normally, we'll talk about adenyl cyclase, but in this case, we've got phosphodiesterase here. It activates that, releases a second messenger, and so on and so forth. And then there'll probably be some questions about the adaptation or recovery phase as well, too. Okay. All right. Now then, where are we here? Let's do it this way. All right. So we could go on for a little bit more. Yeah. But let me take a break for a second and ask you guys if you have any particular questions about any of this before I do that. I want to make sure. I think I will go on a little bit further, but I won't. Um, um, so I'll have more time for a review type thing on Thursday.
So, Dr. Ebert, what you just went over is going to be on our next exam, or the next
Yes.
exam, just that the article that we read.
Um. So think of this as, these two chapters will be on the next exam, and then there's going to be an additional assignment that's going to get counted as an exam, that'll deal with the article. More on that later. I don't know which will come first, but the next exam is going to be a week from today. So yeah.
Okay.
Yeah. But the exam number 5 will just be these two chapters, and this other short assignment will just be the thing, the article. Yeah, I'm still working on that. So yeah. Okay. All right. So I know G protein receptors are a lot, and actually, let me pull up something else here real fast. Maybe. See if I can open it up here. Where's my Poggle? Yes. Okay. So I'm going to share a screen with something else, and we'll take a second here. Normally, we would have an in-class, a thing to work on as well too with this chapter. So there is this nice little exercise on signal transduction. This is available under the exam 5 material. All right, and it's basically, there's a picture in there that will come up at some point, but it's basically a little kind of guided questions about signal transduction in here. We may do something like this as well too, but I want you to kind of look at that and maybe think about looking over those. There we go. There it is. Yeah. All right. But again, you can see here, the eukaryotic example is using epinephrine receptor in our G-coupled protein with adenylyl cyclase and so on and so forth there. I used to go over this in some detail. Sometimes I like this sample, sometimes I don't. We also have an example here with a bacterial cell. A lot of these questions are very short. I want to say simple natured, not necessarily, but they're different from the way I ask questions. So I know I fooled students like, wait, what's he really asking for here? These tend to be very, very straightforward. So example, in model number 1, what's the sphere with the P in it? It's like, okay, it's phosphate. Okay. But this is another good thing to go over as well too. We may do more of this on Thursday, but I just wanted to make you guys aware of that. So like I say, sometime later today, look for some more information about these extraneous assignments. And you're like, Dr. Ebert, stop throwing assignments at us. Good news is we're done with lab, right? Right. Okay. So I'm not going to throw a whole bunch of extra assignments at you, but that's just something there. I'm not going to make that one an assignment, but that's something that's useful to go over, and we'll probably go over the answers to that on Thursday. Okay. All right. So with that said, let's go back to our slides. That was a terrible break, wasn't it? Oops, wrong thing. So much stuff open here. Okay. So for the last 10, 15 minutes, we'll talk about these other types of receptors a little bit. Okay. So we have, enzyme linked membrane receptors. All right. These are our second type that we kind of saw there. And there's not nearly as much detail as with our GPCRs. What we have here is an extracellular ligand binding domain and an intracellular kinase domain. So it's got kinase activity. It will allow it to autophosphorylate. All right. You can see the joke turning in my head there, but it's like, no, it's not worth it, not worth it. But basically, the self-phosphorylation will lead to a conformational change, which will allow it to bind specific target proteins in the cytosol. All right. And then sometimes it'll phosphorylate those. Sometimes, we may see some that have guanylyl cyclase activity, and they'll convert our GTP to cyclic GMP. And we've seen the role of our secondary messenger. So that's kind of what can happen there too. So in this case, we have, should probably actually open this up here, our atrial natriuretic factor, just a particular ligand that will bind here. We'll see the phosphorylation. We'll get the generation of cyclic GMP, and that'll go on and work on other compounds in there. Okay. All right. Some of these are actually soluble. So our nitric oxide activated guanylyl cyclase is one of those as well. Um, but, uh, um, we basically get our protein kinase G. It's like, wait a minute, protein kinase A, B, C, and G. Lots of protein kinases in there. Okay. Our receptor tyrosine kinases. Again, this is part of our modularity. We can take some common domains and just kind of mix and match them. All right, and we'll see here, our various factors that they're going to interact with. A lot of different growth factors and of course insulin response as well too. We'll have these tyrosine kinase domains on the inside. Okay, and then they'll be specific to interact with their particular target proteins. And again, our outside ligand binding domains can vary as well too. Okay, so here we can bind insulin. We can bind all sorts of different things out here. So that's part of that modularity. Just by changing up a few structures in there, you can see our VEGF and our PDGF receptors, vary by two of these Ig-like domains. All right. Take a couple of these Ig like domains and add a leucine-rich section, and you get a different receptor out there. So we're just basically mixing and matching little sections. And I always like to go back to the Lego idea. Here we're taking different Legos, and it's like, oh look, it's a house. Oh look, it's a car. I remember, I think when my son was little, there was a kit where you could build like 3 different things from the materials that were in there. It's just how you arrange them. Okay. So this is an example of our modularity. The best example of a tyrosine kinase receptor is probably the insulin receptor. So of course, insulin gets produced in the islets of Langerhans and the beta cells in the pancreas, and then gets released into the bloodstream upon signaling. And when insulin binds, it says, hey, glucose is coming, turn on the glucose uptake mechanisms, and energy storage function in there as well too. So if you can't make insulin, or if you can't respond to insulin, those are 2 types of diabetes. There's actually a 3rd type, gestational diabetes, that can be brought on by pregnancy, but typically are type 1 and type 2, which used to be called juvenile onset and adult diabetes, but we just call them type 1 and type 2 now for that insensitivity. How does this work? Okay, well, we have our glucose transporters that are basically kind of stored inside the cell in these vesicles. All right, if there's no glucose out there, why keep them out there active? We'll just keep them inside. Keep them inside where they're safe. It's kind of like maybe having a bicycle. You don't want to leave it out in the weather. You don't want it to get rusty. You don't want someone to steal it. So we're going to bring it inside and store it inside. Okay, and then we have our insulin receptor here. Insulin binds to it and activates a signal that says, hey, we're about to get some glucose, and it's good weather, let's go for a ride. Okay, we're going to get some glucose, so that's going to induce these storage vesicles to move to the surface, where of course they can bind to the surface and release the glucose transporters that are in there. And you can see here we have our transporters kind of facing inside. So when they bind, they're now facing out there. They're in the correct orientation. Basically, the outside is the white part, and the kind of salmon colored is the inside of the cell. So we see that how this works out here. This inside of the glucose transporter is always pointed towards the inside of the cell. All right. So we get our glucose transporters out there. Our little invagination kind of flattens out so that we can have access to, say, maybe the lumen of the gut. And we have our glucose molecules that bind to the transporter, and then they get transported inside. as our glucose goes away, I drink my Mountain Dew, and then some time goes by, no more fresh glucose coming in. The insulin production should drop off. Signals will drop off there as well too. We'll have less glucose, so we'll see a signal that'll come in, and we'll take our glucose transporters and make another vesicle, in there that will fuse with this nice little endosome and bring them inside, and we can store them. Okay. Now, sometimes we get things that we don't necessarily want. Sometimes we have old glucose transporters in here, so some of these will bud off and maybe go to a lysosome to be destroyed, but others just go here to be stored. All right. Okay, so let's look at the ligand binding and the signal cascade that gets induced. Here is our binding partner, insulin, binds on the outside. You see this autophosphorylation on the inside. It's inactive. We see these tyrosines, these key tyrosines in here. When it's activated, we see this phosphorylation of these tyrosines in here. So this is as opposed to serine or threonine, we get a tyrosine receptor that's activated in the insulin receptor in there too. Once those are phosphorylated, it allows the IRS to come and audit them. This insulin response something 1 to bind to them, IRS1. Okay, so IRS1 can interact with the phosphorylated receptor on the cytosolic side, and then it becomes phosphorylated as well too. So you can think of like I don't know if you guys ever played some little computer game, Plague or something, basically where you try and design a virus or bacteria that will infect the entire world. It's like, oh no, the ports in Madagascar are closed, but essentially, you get too close, and boom, you get phosphorylated as well too. So IRS1 gets phosphorylated, and then there's a whole raft of proteins, GRAB2, SOS, RAS, RAF, and then the other kinases, these various kinases in here that we see this giant phosphorylation cascade, ultimately resulting in ERK being phosphorylated, which allows it to go into the nucleus and activate these some gene transcription by phosphorylating ELK1, which causes it to dissociate from this other factor in here, which can open up that DNA and allow it to be transcribed. So in this case, we have lots of energy. That's a good time for cell division, right? Because it takes a lot of energy to divide the cell. So we can stimulate the transcription of cell division through insulin, through this whole big signaling cascade pathway. It's a lot, right? It's way too much. All right. So we're like, hey, wait a minute. Somewhere in here. What do we see somewhere in here? Was there a G? No, I'm going back too far. But one of the neat things that happens is you can have crosstalk between the tyrosine kinase receptors and the GPCRs. Okay, so this is an example of integration of signals here. So here we have, this insulin tyrosine kinase receptor. Well, if we have our beta adrenergic receptor here, we're going to phosphorylate it as well too. It's like, I'll phosphorylate you too. Sorry, that's a really obscure and ancient reference there. Anyway, so we see this phosphorylation. If our adrenergic receptor gets phosphorylated, it's going to attract that beta arrestin and be endocytosed, so that we can't get that adrenergic response in here. So basically, our adrenergic receptor, we're thinking like glucagon here. If we're in a high energy state, we're not going to need to release a lot of glucose. If there's a lot of glucose circulating in the bloodstream, we don't need that much more too. Okay. So, if I drink a whole bunch of Mountain Dew and a bear dressed as a clown jumps out at me, I'm ready to go already. I don't need any more adrenaline. I don't know. Certainly lead a weird life here. All right. We can also have what we can call a multivalent signaling cascade. All right. So, we phosphorylate one thing, which phosphorylates another, which phosphorylates another, which binds to another, and all this interaction occurs, and we get more phosphorylation and so on and so forth here. Our multivalent signaling protein cascade is basically lots of stuff is happening. All right. I'm going to get that on an exam answer. Lots of stuff happens. But we see this interaction here, which ultimately results in this ERK kinase being phosphorylated, moving to the nucleus, phosphorylating some DNA binding proteins, that allow that DNA to be accessed when they leave. Sorry, when they bind, which stimulates the transcription of the mRNA that is involved in cell division. Whew, that's a lot. All right. The other thing is multivalent adapter proteins. You're like, what? There's more? And I feel like I should be like the guy selling stuff on TV. But wait, that's not all. Act now, and we'll throw in some multivalent adapter proteins. So some of you may have had some lab experience. You worked in here where you talked about SH2 or SH3 domains. But again, we just have all these little protein domains, and we can kind of mix and match them to see what's happening. It's like, for example, in signal regulation, we have our socks domain here. Okay, we're wearing our cool socks today because it's Tuesday. I don't know. But we can have all these different types of domains in here, and again, this fits in with our modularity. If we switch out a few domains, we can get a different reaction, a different interaction in there. All right. And then, the book likes to talk about SH2 domains a little bit. SH2 are very well studied. We're not going to talk about them too much. The big thing I want to take away from you guys is we can have this scaffolding protein here as well too. Okay. So the scaffolding protein, KSR, essentially holds a bunch of these in close proximity to each other. Okay. So, we have our phosphorylated RAF, our phosphorylated MET kinase, our phosphorylated ERK kinase, and we get this strong output. Okay, if we phosphorylate the KSR, all right, we're going to kick off the RAF, so we're not going to phosphorylate MEK or ERK, so we're going to much weaker output. Our scaffolding protein is basically an example of a multivalent adapter protein. It holds everything in close proximity, but if it gets phosphorylated, it boots them away. They're not in close proximity. So what we see is this quick response as everything binds to the scaffolding protein, and then the scaffolding protein helps regulate that response and turn it off. Okay. If we mutate this, okay, so that our KSR lacks a phosphorylation site, then we see a constitutive activity. All right, it's always on. We always get this big response. So whatever our input is, we get a strong output. There's no adaptation because there's no phosphorylation site to inhibit the collection of these different compounds. I hope that makes sense. I like this picture. I like seeing what's going on here. We have all this stuff that's going on. But eventually our scaffolding protein gets phosphorylated and we slow it all down. Okay. All right. Okay. So that's probably enough for today. We will pick up with all of this. We only have about 15 more slides to go anyway, but we'll pick up with ion channels on Thursday and integrins and nuclear receptors. And then we'll wrap up. We'll summarize the entire semester. Know everything. No, okay. But yeah, so that's a lot. But that's enough for today. That's good. I always like to have an extra day built in or something so I can do other stuff. So, I've cut out all the fun stuff this semester, and you guys are like, yeah, you sure have. And none of this was fun. Okay. Hopefully some of it was fun. But that's what we've got for today. Like I say, on Friday, we'll finish up. We'll go over maybe that worksheet, and we'll talk about the assignments and the exam that's coming up as well, too. We're almost done with our semester. Yay. So have a good early part of your week.